Hello students, welcome to my channel. In this class, we will learn about the DNA probe which is used in microarrays. What is DNA probe? And thousands of DNA probe are fixed on the solid surface, as we learned in last class. That probe are non-DNA sequence which are fixed on solid surface. Each probe has different sequences. Means each probe are different sequences. That's why we are using different thousand of sequences. If there is same sequences, that will use for the check the expression level. In this, we are using the different gene expressing. So there is the difference. So that's why we are using all different probes. No single probe is similar to each other. Okay. Each probe represent one gene. That's why it represent a single gene is equal to single probe. So 1000 probe means 1000 genes are there, which will use to calculate, determine or detect the number of gene expressing in our sample cell or sample material, whichever you consider. Type of probe. Which type of probes we use most of the time? That is synthetic oligonucleotides, which are from 10 to 25 in nucleotide in number, and cDNA means mRNA to converted into the cDNA with the help of reverse transcriptase, and PCR product, which are making many copies of cDNA, so it can be, if there is so little in amount, that could be possibility to miss the base pairing. If there is number high, then it will be have higher chances to find out the complementary sequence in that probe. So that's why these type of we use probes. So type of DNA microarray based on density. On the basis of density, we categorize the microarray. There is two type mainly, low density and higher density. What is low density? Probe spot on glass slide and nylon membrane. When our array use less amount of, as I said, 1000. If there is not 1000, we using little amount. Then there will we use the solid surface we use will be glass slide or nylon membrane, not the silicone. And there will be lower density, then we are using few spots, not many in number. Higher density array, how it is different? Here we are using silicon one as a solid surface, and their spots are 10 power 6, and which is in 1 to 2 centimeter area. Like this is the solid surface. Only this. It doesn't mean that I am representing on a screen, that's why it is a small. No, this is the 1 centimeter. Even it is more than 1 centimeter. So this much area have this much of spots, like very small spots are there. We cannot even see from our naked eyes. This is used in higher density. What is then the microarray and macroarray? What is the difference between these? When spots are spot size, it depends. Features क्या है? What is the features on the basis of these are named? Spot size, number of spots, and density. If there is low density, it will consider macroarray. We use macroarray, which have more than 300 micron in size, and spots are few in number. If we are using this much of our solid surface and we are making it size large, then obviously we have few spots. If there is less than 200 micron size, then we use high density and high number of spots, like thousands of spots. Important points are those we are already discussed. A single DNA microarray chip contains thousands of spots, and each spot one gene. Each spot is considered a one gene. Remember that. So this much of spot will almost entire genome. So that's why we call it a macroarray. So it is using high density, and this is the size of the spot, and if we are using this, then it will not named as macroarray. Remember that. Okay, in next class we will learn about type of DNA microarray. DNA microarray types, and what is their basis of categorization, we'll see in next class. Okay, bye bye.